आज मैं यूट्यूब लाइव आया हूँ। कितने अटैच हैं ये क्लियर नहीं हो पा रहा है। मैसेज में कुछ आ नहीं रहा है। लाइव समर लाइव it is not clear from the screen that the students are linked or not. Give some messages to identify that the students are linked here. One message I received, that is Sunil Kumar. Here, today we are going to discuss or interact on the topic biochemical micromolecules. That is a very important topic for the CSIR-NET examinations. And like similar examinations, and sometimes the students did wrong, selected wrong choices for very simple questions due to the students are not give more emphasis on the basics of the biochemical molecules, particularly associated with the microbiochemical molecules. And in the biological system, we discussed earlier that during the origin of life, primary biochemical molecules were developed or evolved either by the intermediate SCN or through activated acetate entered into the analytic cycle, and in this way they obtained energy from the geochemical processes. But during the course of evolution, the biochemical molecules were primarily evolved to fulfill the different functions of the biological system, as well as to establish their structural energetics. So, the most important point here to study the basics of the biochemical micromolecules and a structure particularly associated with the configuration in the biological system. And in the biochemical molecules, fundamentally we considered amino acids, monosaccharides, carboxylic acids, vitamins as the we interact on the three basics biochemical macromolecules, that is amino acids, monosaccharides, and carboxylic acids in the living system known as fatty acids. The components are important, but they are the isomerism, particularly the stereoisomerism, are more important for the point of view of examination, particularly with the NET. But when the viewers are associated for the preparation of other examinations, like different medical examinations and other biotechnological examinations for the biotechnology entrance, the macromolecules are more important. And at first, I am going to discuss on the amino acids. And the viewers are requested to keep some notebooks and pens and write if they feel necessary. The amino acids are most important for the biological system because they are polymerized to form proteins. And proteins are responsible for regulation of all enzymatic activities because all enzymes are proteins, as well as they are structural components of the living system, even the biological membranes and all the components of the all the components of the cell organelles. If we want to study the amino acids, the more than 200 amino acids are observed in the living system, but they are observed in the catabolic pathway or as the intermediates of the biochemical metabolic activities and biochemical reactions. But if we want to study the amino acids involved in the protein synthesis or coded by the mRNA, then they are only 20 in number. These 20 amino acids are significant for the synthetic nature of the proteins, and the 20 amino acids are associated with for the formation of different configurations of biochemical molecules and structures. If we divide the proteins on the basis of number of amino acids, then it is referred as peptides, peptones, and polypeptides. If we observe up to 20 residues, then we said they are peptides. 20 to 50 residues are present in the peptones, and when more than 50 amino acids are present in any chain, then we say it is the polypeptide. But it is the nature of the proteins, which we will again discuss in either in classroom or in the online classes or on the online live classes. But at present, we concentrate on the micromolecules only. If we study the amino acids, then structurally amino acid is formed by alpha carbon, and the alpha carbon is associated with two functional groups, carboxylic functional group and amino functional group. And one bond is neutralized by the hydrogen, and one bond is represented by R, that is the alkyl group, which represents variation from amino acids to amino acids. That means here the one carbon is associated with one amino acid, one carboxylic acid, one alkyl group, and one hydrogen. In this way, the common structure of the amino acid represents association of both basic and acidic group with the same alpha carbon. Acid has ability to release the H plus, and base has ability to accept the H plus. In this way, a reorganization is observed in between carboxylic group and amino group associated with the alpha carbon, and it represents in an ionic form. And the ionic form of the amino acid is referred as deuterium ion. That means in the biological system, the amino acid can be present in two forms, in the neutral form and in the form of the deuterium ion. It depends on the physical conditions of the cell and cell environment, or the physiological condition of the body or the environment of the body, which we will discuss later on. And here we then see what is the nature and variation of the R, that is the alkyl group. And on the basis of the variation in alkyl group, the amino acids are of 20 types. Then the amino groups associated with the the alkyl group associated with the particular amino acid is responsible for the secondary bonding or non-peptidic bonding. Hence, it is responsible for the configurational setup of the proteins. So, It is most important part for the study of the configurational organization of any proteinous molecule. If we observe the common structure, which I am telling just, that carbon is associated with amino group, carboxylic group, one alkyl group, and one hydrogen. But the common structure is not followed by two amino acids. One is glycine and one is proline. Glycine R of glycine is replaced by H. That means R is associated with, C alpha carbon is associated with 2H, one COH carboxyl group and one amino group. In proline, the N is present in imino group, not in amino group. So, the nature of R is not followed the common plan of the amino acids in glycine and proline. Secondly, if we study the 20 amino acids, they are at first divided into three groups. One, neutral amino acids. Second, acidic amino acids. And third, basic amino acids. At first, you understand the difference between neutral amino acids, acidic amino acids, and basic amino acids. If hydroxylic carboxylic group, that is COOH, is present in R of common structure, then the amino acids are acidic. If amino group is present in R of amino acid, it is referred as basic amino acids. For example, the two amino acids are acidic. That is aspartic acid and glutamic acid. Because in addition with the alpha carboxylic group, one carboxylic group is also present in R group, alkyl group. The basic amino acids are three, that is arginine, lysine, and histidine. In arginine and lysine, amino group is present in their R. In histidine, the amino receivers, electron H plus receivers group is present in the histidine. That means these three amino acids, lysine, arginine, and histidine, are basic. Now we come on neutral amino acids. In neutral amino acids, the alkyl group is neither contain the SD group nor contain the basic group. That means they have no power to donate the H plus ion or receive the H plus ion or donate the OH minus ion or receive the OH minus ion. That means they are neutral. But in some alkyl group, slightly electropositive or electronegative components or elements or groups are present in some neutral amino acids, not in all. And they are responsible for the development of polarity. Hence, the neutral amino acids are further differentiated into neutral nonpolar and neutral polar amino acids. At first, we consider the neutral polar amino acids. In neutral polar amino acids, if we observe the structure of the neutral polar amino acids, it is very easily you analyze that they are the neutral and polar, because the polarity, for the development of polarity in amino acids, OH or SH groups are present. Either OH or SH groups are present in two conditions more: the asparagine and glutamine. The asparagine and glutamine amino groups are present, that is reduction of the OH COH group from the aspartic acid and glutamic acid. That means the two amino acids, methionine and cysteine, has sulfur SH S group, and they become polar. Serine, threonine, tyrosine. Serine, threonine, and tyrosine have OH functional group, and the OH is responsible for the development of polarity. That is 5. 2 SH containing, they are sulfur containing, 3 OH containing, 2 asparagine and glutamine, 5 to 7 amino acids, and 1 glycine, which is slightly polar, due to the presence of H instead of R. That means the 8 amino acids are neutral but polar. Neutral but polar. Rest 7, that is alanine, valine, leucine, isoleucine, proline, phenylalanine, and tryptophan. These 7 amino acids are non-polar. Then these are the basic chemical nature and structural variation of the different types of the 20 amino acids. Again, in some questions and in sometimes question is asked after giving some value, that is the atomic mass unit and total weight of the protein, and setter asked the number of amino acids. And it is very simple. If you understand properly, that it is very simple. And it is difficult for anyone or any student to remember the molecular weight of all the amino acids. Then you count the molecular weight by putting and summing the values. You are simply, you always simply remember that the average weight of the atomic weight of the amino acid is 132 if we collectively take the average of amino acids of all the 20 amino acids, then we remember only the 130, 130, 16, 36, 136 of the value of the amino acids. 138. 138 is the average value of the amino acid. And when two amino acids are combined by the polypeptide bond, and during the formation of the polypeptide bond, carboxylic group of the first amino acid and amino group of the subsequent amino acid are associated and form a bond by the removal of one H2O. And here the value of H2O, molecular weight of the H2O is 18. So we subtract the 138 minus 18, that means the 120. And if any question asked the total number of amino acids by giving the total atomic weight of any polypeptide chain, then you simply divide the total value by the 120 and you get the approximate number of the amino acids. So it is the simple to calculate if asked in the examination. And the amino acid is again polymerized, and we will discuss just the stereoisomerism in this session. The second important micromolecule is the monosaccharides. The monosaccharides are naturally occurring polyhydroxyaldehyde or ketone. Polyhydroxyaldehyde or ketone. And commonly the ratio of carbon present is same as the ratio of water in the molecular formula. That means if 1 unit of carbon is present, then H2O is also represented in the form of 1 unit. That is CS2O. If C is 2, then H is 4, O is 2. Like this. And in nature, the monosaccharides or saccharide units are abundantly observed, abundantly present. Commonly, just I am asking that, it is the polyhydroxyaldehyde or ketone. That means the functional group present in the monosaccharides are either aldehyde or ketone. The aldehydic form is referred as aldose and ketonic form is referred as ketose. Commonly, we observed the monosaccharide in the biological system from the 3 carbon. That is the triose. And in the triose is commonly observed as a metabolic intermediate. Naturally, it is not observed. And the commonly observed trioses are glyceraldehyde and dihydroxyacetone. When the carbon is 4, it is referred as tetrose. And the common tetrose is erythrose and erythrulose 2. Then pentose. Pentose is most important after the hexoses. And of course, most important than the hexose, because the pentose is responsible for the formation of genetic material and nucleic acids. And that pentoses is ribose and ribulose. Then hexose, and a number of hexoses are present. That is mannose, arabinose, glucose, fructose, fucose, galactose. A number of monosaccharides are, hexoses, monosaccharidic hexoses are present, which have the 6 carbon. Now, the monosaccharidic unit commonly monosaccharidic unit is unite to form the disaccharide. And disaccharide is present in nature. I am discussing here the disaccharide because if we observe the biochemical parameters for the differentiation of the micromolecules and macromolecules, then conventionally we consider that the atomic mass unit up to the 1000 are included in the micromolecules. And when the atomic mass unit or weight is more than the 1000, up to the 10 to the power 9 are included in macromolecules. So here I am discussing the disaccharides and trisaccharides and tetrasaccharides. The disaccharides are formed by the combination of 2 units of monosaccharides. That is, one is the maltose, commonly observed in the nature. Maltose is formed by glucose and glucose. Then trehalose. Trehalose is also formed by the glucose and glucose. Then lactose. Lactose is formed by glucose and galactose. Then sucrose. Sucrose is formed by the glucose and fructose. Here, I am asking that the maltose and trehalose both are formed by the 2 units of glucose. Maltose is formed by the 2 units of glucose and trehalose is also formed by the 2 units of glucose. Then what is difference? And we will discuss just, हम लोग इसको just discuss कर रहे हैं, because they vary on the basis of the nature of bond, and bond is varied due to the variation in the stereoisomerism. Hence, the stereoisomerism is most important part for the biological system. Then we comes on the trisaccharides, and trisaccharide is formed by the 3 units of monosaccharides. The trisaccharides are monotriose. Monotriose means glucose plus glucose plus glucose. Ribinose. Ribinose is formed by the galactose plus 2 units of ribose. rambose is the isomeric form of the glucose. Then raffinose. Raffinose is formed by the fructose plus glucose plus galactose. Genetobiose. Genetobiose. Genetobiose is formed by the fructose, glucose, and glucose. Now the tetrasaccharides. Tetrasaccharides is rarely observed in the nature. Tetrasaccharide is not commonly observed. But two tetrasaccharides are commonly observed. One is estachylose. Estachylose is collected from the plant Stachys tuberifolius. Tuberiforms. Then escaldose. Escaldose is formed by the bulb of garlic and onion. Escaldose is observed in the bulb of garlic and onion. Then these are the common nature of the monosaccharides. Then we observed commonly the carboxylic acid, popularly known as the fatty acids. We observed popularly as the fatty acids. The fatty acids, which are observed in the living system, are long chain hydrocarbons with terminal carboxylic group. It is the long chain hydrocarbons with terminal hydroxylic group. In the last of the carbon, the hydroxyl group represent, and that associated is known as the alpha, then the beta, then the gamma, then the delta. Then there is a nomenclature is based on the Roman symbols, alpha, beta, delta, gamma, delta, like this. So, if we observed the energetics in later chapters, बाद में उसको हम लोग देखेंगे, then we easily observe that the fatty acid is high energy producing component. इससे बहुत ज़्यादा energy produce होता है, which we will see in later part, in later classes or online like this. Here, over 70 types of fatty acids are observed in nature or isolated from the various tissues and cells. But the fatty acids are commonly divided into the two parts: the saturated fatty acids and unsaturated fatty acids. It is very simple. In the saturated fatty acids, the pi bonds are absent. That means the secondary and double and triple covalent bonds are absent. But in unsaturated fatty acids, the double or triple covalent bonds are present. Commonly, the double bonds are present due to the neutralization of the valency between the carbon and carbon. Commonly, it is formed between the carbon and carbon. So there is a maximum possibility for the formation of the double bond between the carbon and carbon. In the saturated fatty acids, some common are lauric acid, myristic acid, palmitic acid. All these are the unsaturated fatty acids. And the unsaturated fatty acids commonly represents the CN, number of carbon is represented by N, and the H2O number is represented by 2N plus 1 and one carboxylic end, that is COOH. That means it is commonly represented by CN, H2ON plus 1 and COOH. It is the common uh, formula for the saturated fatty acids. But when we observed in the unsaturated fatty acids, each double bond is responsible for the reduction of two hydrogen. This is common principle of the chemistry that when the one double bond is formed, then it is responsible for the removal of the two hydrogen. That means if we observed in the one double bond in any fatty acids like in the palmitic acid and oleic acid, in this form one double bond is present, and that means the two hydrogen is reduced from the common formula. And when we observe the two double bonds as in the linoleic acid and arachidonic acid, then the four hydrogen bonds are reduced from its common formula. Now, the it is the common formula. The some part is also included in the micromolecules, like some energy producing chemicals and nucleotides. But I am not included the energy rich compounds and nucleotides in this session because I will give you a detailed lecture or detailed outlook, or I will provide a detailed teaching concepts on the in the next class. I particularly in the classroom. But if we observe, you all know that the nucleotides are formed by the phosphoric acid, sugar, and nitrogenous bases, which we will discuss in the later. The high energy compound, if you observe in the high energy compound, the high energy compounds are of three types. All are the micromolecules, which we are discussed in very brief. The high energy compounds are of three types. That is the thioesters. First is the thioesters. If you have any some, if you have the some idea of the glycolysis or Krebs cycle or energy production system catabolic activities, then you easily understand that the three components are commonly used for the energy production in the living system. That is the thioesters, anhydride forms like the ATP, and the phosphoenols group. So these three are the high energy components which we will discuss in the later on. And second part is the nucleotides, which I will also discuss in my next class. But most important is today is now the stereoisomerism. As I think that you all have a fundamental knowledge of the micromolecules of the amino acids, saccharides, and the fatty acids. Because the fundamental understanding is necessary for the proper understanding of the isomerism and nature of the bonds formed later on. Now, we come on the stereoisomerism. In the stereoisomerism, we observed, we studied, you all studied in previous classes that the chemicals have some physical properties, some chemical properties, though the compounds have some physical properties, some chemical properties like this. But in the biological system, the physical property and chemical property are not as important as the biological properties. And here, the micromolecules, the biological properties of the micromolecules are determined by the stereoisomerism of the biochemical molecules. And if we observe, the stereoisomerism, then stereoisomerism represents that the same molecular structure, same conformational structure, but different properties are included under the study of stereoisomerism. That means the biochemical molecules, all the biochemical molecules, or the stereoisomerism, all types of the stereoisomerism are more or less included in the biological system and have the significant. Why significant? Because on the point of view of the energetics. I am repeatedly said that in the biological system, most important is the energetics. Energetic is the most important. And when the biochemical molecules were originated in before 3 billion years ago, sorry, 2 billion years ago, they are evolved continuously. They were evolved continuously, or they are evolving continuously to minimize the level of energetics and get the more stability, structural stability. And why so? We are saying that the evolution is a continuous process, not only in the living system, but in the biochemical system also. And the isomers, which we study the isomers, then isomers are simply divided into the structural and stereoisomerism. Structural isomerism is also known as the conformational. That is conformational and configurational. Stereoisomerism is also known as configurational. Then the stereoisomerism is again divided into enantiomers and diastereomers. Enantiomers and diastereomers. Enantiomers means the optical activity, determined by the optical activity. I am just going to describe in the present session all these things. Enantiomers, that is the optically active, and diastereomers, that is configurational. That is configurational. Again, diastereomers is divided into geometrical and conformers. Geometrical and conformers. That means the enantiomers is represented by DL. I am just saying because you understand what I am going to describe. Enantiomers is D and L. When we say that the dextroform and levoform, they are the enantiomers. Second, diastereomers. Diastereomers is the two types. One is the geometrical, that is the cis and transform. Cis and transform. And third one is the conformers. Conformers means the torsional arrangement of the bond among the molecules of any biochem elements of the any biochemical molecule. And this form, they are the different forms like the boat form, chair form, like this. Just I am going to describe. Why so I am still here? I am not going to explain the DNA, because in the DNA, in the organization of the DNA or in the organization of the RNA, the conformers form, the forms represented by the conformers are more important. But here we are discussing on the stereoisomerism, and first we discuss on the in enantiomers. Then what is enantiomers? In enantiomers, if asymmetrical carbon is present in any biochemical molecules, asymmetrical carbon. What I am saying? Asymmetrical carbon. Asymmetry means the carbon has the 4 valency. Carbon has the 4 valency, and if the all valency of the carbon is neutralized by different components, different groups, then the carbon represents asymmetrical nature, and it is referred as chiral carbon. It is represented as chiral carbon. Then it is better to say that in the chiral carbon, all the 4 valency is fulfilled by 4 different functional groups. If we observed in the amino acids, the one carbon is associated with the COH, one is associated with the NH3, one is associated with the R, and one is associated with the H. That means the alpha carbon of the amino acid, except in the glycine and proline, represent the chiral nature. That is the asymmetrical. If we observed in the glucose, then one carbon is associated with the aldehyde group, one bond is neutralized by the H, one bond is neutralized by the OH, and one bond is neutralized by the C4H8O4. That means here the first carbon is associated in the aldehyde is associated with the oxygen, COH. That means is associated with the oxygen. That means it is not chiral. But in the second carbon, one is associated with the aldehyde, one is associated with the H, one is associated with the hydroxyl group, and one is associated with the remaining part of the R of the glucose. That means the second carbon represents the chiral nature. And when the chiral carbon is present in any biochemical molecules, then they become optically active and represents D form and L form. D form means in the plane polarized light is reflected in the right hand direction, and in the LEBO form it rotates in the left hand direction. That is the D form and L form. Now the chiral carbon present in the amino acids, the chiral carbon present in the monosaccharides, the chiral carbon present in the fatty acids, and that chiral carbon are responsible for the optical activities of the biochemical molecules. And if they represent the variation, these components are referred as enantiomers. These biochemical molecules are referred as enantiomers with each other. And the enantiomers, not in the stereoisomer, in the condition of the enantiomers, the enantiomers are having enantiomers, same enantiomers having same physical and chemical properties. Same physical and chemical properties, but they differ in the biological properties. And not only differ in the biological properties, but they differ on the bonding natures also. When they polymerize or when they want to get the secondary configuration in the condition of the protein, they represent the variation. For example, if we observe the chiral carbon, it is very easy to consult with the chiral carbon. And if we have any problem in the determination of the chiral carbon, then you give me comment in my next in the present video. Then I will differentiate or give you proper knowledge for the determination of the chiral carbon in the next class. And if we determine the chiral carbon, then the number of enantiomers is determined by the 2 power n, where n represents the number of chiral carbon present in biochemical molecules. And likely here, for example, just I am saying that in the amino acids, the alpha carbon is the chiral carbon, because all the 4 functional groups are different. But not only here. If you want to determine the chiral carbon, you have the proper understanding and proper knowledge of the structure also. Suppose in the amino acids, we consider the valine. In the valine, the R is represented by CH, CH3, CH3. That means here the one alpha carbon and one beta carbon both becomes chiral. That means the number of isomers is determined by optical isomers or enantiomers is determined by the proper understanding of the chiral carbon. And if we consider in the natural availability, then the amino acid naturally observed in L form, in LEVO form. Commonly the amino acid observed in the natural form is LEVO form. That means the LEVO amino acids are commonly observed. Sometimes the LEVO rotatory or the enantiomers, nature of the enantiomers has used in the development of the pathological markers or the defects in the biochemical pathway. For example, I am going to give you a best example. That just I am saying that the amino acids are present in the L form in natural condition. But if we observed D-serine, that is the dextroserine, in the urine of any person, or if we analyze the D-serine in blood of any person, then the presence of the D-serine in blood is strongly related with the deficiency of the glomerular filtration rate. And nowadays, it is useful marker for the estimating of the function of kidney. So, in the field of research, the optical isomerism, or in the organization, or in the development of the drug designing, in all these fields, the isomerism or stereoisomerism of the biochemical molecules are most important. Now, we study the amino acids. In the second stage, if we study the monosaccharides, particularly in the hexose, commonly available and more abundantly observed monosaccharides are hexoses. And then the hexoses represent more enantiomers and enantiomers. And then, because in the monosaccharides, the chiral number of chiral carbons are multiple. If we study the hexose, then the second carbon is chiral, third carbon is chiral, fourth carbon is chiral, fifth carbon is chiral. All are chiral. If you analyze this, then all are the chiral carbon. Then there is more chances of the enantiomers available in the biological system or in the living system. And here, the monosaccharides mostly observed in the natural condition are in D form, in dextrorotatory form. And the dextrorotatory form, it is conventionally to analyze the dextrorotatory form and a levorotatory form from the configuration present in the book or present in any paper. If the farthest hydroxyl group from the functional group is on the right hand side, then it is referred as D form or dextrorotatory. And if it is present in the left side, then it is referred as the levorotatory form. That means the D form and L form can be also easily determined on the basis of the OH, distant OH from the functional group on the paper or in the any diagram. Then I am just saying that the monosaccharides are present in the aldose and ketose form. In nature, the fructose is only natural occurring ketohexoses. It is widely observed. It is also referred as the fruit sugar. But the fructose present in the honey, it is most important for the metabolic activities and for the proper understanding of the stereoisomerism in the biological system. But the fructose present in the honey is levorotatory. It is not dextrorotatory. Why so? It is referred as levulose. Of course, it is fructose, it is ketohexose or fructose, but it is stereoisomerically different. It has the different property and it has the different name, that is the levulose. Of course, it is the fructose, hexoketose, but it represents the levorotatory nature. And when it represents the levorotatory nature, it is metabolically less active. Now, we observed that the glucose is present both in the linear chain or in ring form. In ring form, if we observe it is in ring form, can be it is represented in that the first carbon is associated with the O and the fifth carbon is associated with the O. That means the first and fifth carbon are associated with the O to form a ring. And here in the ring form, it has two isomeric forms. And in the biological system, the sugars are majority the glucose are present in the ring form. And on the first carbon, the OH group or H group is associated with the first carbon, second carbon, third carbon, fourth carbon, and fifth carbon. That means here the OH of the rotation of the OH on the first carbon is responsible for the determination of glucose ring into alpha and beta forms. When OH of sugar is in lower direction, then it is referred as alpha form, and when it is in upper planar, then it is referred as beta form. That means in the ring form, glucose is present in alpha form and beta form. And these alpha and beta forms, their configurational structure of the alpha and beta form of the sugar are especially important when the sugars bind together to form polymer, which we will discuss in the macromolecules. These bonding natures are different. The nature of the alpha bonding and beta bonding are different. Why so? Sometimes you in book, you observe that if alpha glycosidic bond and beta glycosidic bond, due to the stereoisomerism and planar differentiation of the OH of first carbon, and of course the fifth carbon of the other. Now, when we consider the fatty acids, optical isomerism of the fatty acids, then it has less chance of the configurational changes because it has the long carboxylic group, but the nature and orientation represents it into the cis-N transform. That is the geometrical isomerism. They represent the geometrical isomerism. And due to the variation in the cis-N transform or geometrical isomerism nature, they represent differences in the biological properties or properties in the living system or in the chemical system. In cis form, if we consider the fatty acid in cis form, they have a kink in their chain. Kink in their chain, that means it is not linear. If you study in the cis form, then the fatty acids represent the kink form, K-I-N-K, kink form. That means it represents the king form, and due to the cis nature or a structure, kink structure, they do not pack together very easily. They do not pack together very easily, and due to the not packing easily, their boiling point is very low. Such fatty acids have the low boiling points, and the non-sticking, it is likely to say that the sticking formation of the sticks, that is the non-sticking nature. The boiling point is low, and it has decreased LDFs. LDF, the full form is the LDF is London dispersion forces. London dispersion forces. That means the forces responsible for the thickening of the or compacting of the fatty acids. And due to the decreased LDF, the value boiling point of cis fatty acids are low, and due to the low value of the boiling point, they are commonly present in liquid state. They are commonly present in the liquid state at a room temperature. But in the transforms, when fatty acids are present in the transforms, commonly the fatty acids present in the transforms represent more double bond and triple bond. Double bonds, one or two places. That is the trans fatty acids. And trans fatty acids are have more increased LDFs. Trans fatty acids have increased LDF. That is the forces, London dispersion forces are more to stick, and they stick together. And due to the sticking and compacting of the trans fatty acids, they have the high boiling points or melting points at the room temperature, and they are more harmful, and they are sticked on the blood vessels to increase the blockages. And particularly, what you use? The hydrogenated banaspati oils, that is the dalda. All are the trans fatty acids. And then the trans fatty acids are, generally the trans fatty acids are solid in the room temperature. And the these forms are represented the trans and cis, that is diastereoisomers or components of the geometrical isomers. The third one is, because the third one and the important part is the conformers, which we will the conformers we will discuss during the organization of the proteins, and particularly in the amino acids in the DNA and RNA in the next class, or in the further class, whensoever it will. Then the conformers, and here the conformers means the torsion of the elements associated in the hexoses or biochemical components. That is the bonding angle and torsions. And due to the torsions in the conformers or the gaining of the conformer uh, conformational isomers, of course it is the conformational isomers are most important point for the development of the drugs, metabolites, and of course the configuration, proteomics, genomics, like this. Because the conformational isomers are divided on the basis of the torsional stress, and they are of the commonly five types. One is planar, second is half chair, third one boat, fourth one twist boat, and fifth one chair. These are the conformational isomers. That means glucose also represents conformational isomers. Many drugs represent the conformational isomers. Fatty acids represent the conformational isomers. And the two things which are responsible for the conformational isomers are uh, energy and stability, configurational stability and energy. And here the most important factor, that is the more energy if any biochemical molecule, conformational isomer of the biochemical molecules has uh, high energy, then it represents the least stability. And if it has the least energetic value, then it represents the highest stability. Why so? The For the gaining of the highest stability in the biological system, the biochemical molecules uh, wants to gain least energetic level. That is main point, and that is responsible for the conformational isomers. And the conformational isomers we will discuss in the later class. Then, if we observe the bonding nature, of course, we are describing here the role of the bonding natures due to the variation in the isomers. Then, the bonding nature is bonding is the primary bonding, that is the peptide bond in the proteins. In the proteins, the peptide bond is formed by carboxylic group of first amino acids and amino group of the second amino acids by the removal of one molecule of water. In the glucose, it is formed by the removal of the one molecule of water. In between, OH of the one carbon of the one first glucose or first monosaccharides, and OH of the any carbon of the second monosaccharides, that is the glycosidic bond. Then third one is the ester bond. In the lipids, it is formed by the, because the lipids or the fatty acids are the formed by the polymerization of the glycerol and the fatty acids. That means the ester bond is formed here. And in the esteroid, that is the another point which we will discuss in the later on. Not I am going to discuss here because time is also not permitted me to discuss more on the topic. Now, in addition with these primary bonds, they are referred as the primary bonds. Some secondary bonds are also present, which is responsible for the configuration of the more polypeptide, configuration of the biochemical macromolecules. And the common bonds which are we observed in the biological system. They are the non-covalent bond. First one, that is the non-covalent bond. The non-covalent bond, in addition, all these three, the peptide bond, glycosidic bond, ester bond, of course, phosphoester bond, and disulfide bond. In addition with these, except these, all are the non-covalent bonds interaction, and they are responsible for the interaction between the biochemical molecules. The first one we observed in the biochemical molecules is the hydrogen bond. And hydrogen bond is formed between the neutral groups, that is hydrogen of one group and oxygen having the lone pair electron of the another group. And it has the 8 to 21 kilojoule energy per molecule energy. In second, between polarized R group of the one peptide and another the component of the R group of the another peptide. Here also it is the 8 to 21 kilojoule energetics. Ionic interaction. The ionic interaction has both types, attractive and repulsive. Ionic interaction may be attractive and may be repulsive. In attractive, the positive and negative charged molecules are present. When positive and negative charged molecules are present, then it is responsible for the attraction, and it has 42 kilojoule energetics. Repulsive, when both positive are present, then it is responsible for the repulsive. It has the minus 21 kilojoule. Then hydrophobic interaction. And in hydrophobic interaction, the nature bonding energetic value is the 4 to 8 kilojoule. And in last one is the van der Waals forces. It is the weakest force. And van der Waals forces is formed in anywhere when it forms the partially positively charged and partially negative charge in the air. And it is the very less energetic, that is the 4 kilojoule. Now, I am thankful to all those students who are connected here, and now possibly the time is going to terminate by 3:30. So I am very much thankful to all those viewers or students who are connected here. And if anyone wants any question, then give me message, or I request to viewers to give comment if anyone wants any query or any further explanation of the facts, because on the online it is sometimes it becomes difficult to explain all these things in classwork mode. So I am thankful to all those, and very thankful. Thank you to all. Have any question? No. Now I am going to terminate the